situations or cases confronted by OSIS during inspections of sites. The purpose of the case studies is to emphasize the principles learned during the workshop. The instructors will present detailed scenarios based on OSIS inspectional experience. This will be followed by questions for considerations by the participants. Then the participants will be encouraged to share their thoughts and answers. The instructor will triage the answers and also provide the correct answers at the end. We aim for a very interactive sessions, so don't be shy about typing into the Q&A chat pods. So for our first case, we have a good laboratory practice or GLP case study, which will be presented by Dr. Lynda Lanning and Dr. Zhou Chen, both who gave us excellent presentation yesterday, but here's a reminder of who they are. Dr. Lanning is a member of the GLP team in OSIS. She conducts and evaluates inspections of non-clinical studies submitted in support of pharmaceutical development. And Dr. Chen is a team lead for the GLP team in the Division of New Drug Study Integrity in OSIS, and has experience working as a pharmacology/toxicology reviewer in CDER and performing GLP and animal study rule inspections. And now I will turn it over to our instructors.
Hello, and thank you for joining this session today. My name is Lynda Lanning, and I'm a member of the GLP team in CDER's Office of Study Integrity and Surveillance. Today, along with Dr. Zhou Chen, we'll be presenting some cases regarding good laboratory practice studies. Disclaimer: the opinions and information in this presentation are those of the author and do not necessarily represent the views and or policies of the U.S. Food and Drug Administration. During this presentation, we'll briefly describe the most commonly cited observations from GLP inspections. We'll go into a bit more detail regarding the handling of protocol deviations and issues encountered in GLP study reports translated into English, as we describe a few case studies. The most common noncompliance observations from GLP studies are QAU failed to determine that protocol and SOP deviations were made without proper authorization and documentation. Study director failed to assure that all experimental data were accurately recorded and verified. SOPs have not been established. QAU did not review the final report. And issues around the archives and formulation analysis. Today, we'll be discussing cases with issues around the handling of a protocol deviation and report translation and data accuracy. In case 1, I'll present some issues around the handling of protocol deviations. This study is a GLP-compliant toxicology study with 4 groups of experimentally naive cynomolgus macaques, 3 groups receiving the test article, and 1 control group. The test article was administered by a slow intravenous bolus twice a day for 4 days, followed by once a week for 24 more days. There was a 14-day untreated recovery period. Assessments for this study included ophthalmology, ECG, body weight, clinical observations, clinical pathology, necropsy, and histopathology. On study day 2, high-dose females were showing clinical signs of toxicity after the morning dose. After the evening dose, the clinical signs of toxicity increased in severity for 3 of the 5 females. There were no males or lower dose females showing clinical signs at that time, and the controls remained normal. Clinical pathology of affected animals showed signs of dehydration. On study day 3 in the morning, the high-dose females again showed clinical signs of toxicity after the morning dose, and the severity of those signs increased over those noticed on day 2. High-dose males showed similar but much less severe clinical signs of toxicity, and no mid or low-dose males or females or any control animals showed any similar clinical signs. So it seems that the toxicity is limited to the high-dose group so far. And the affected animals showed clinical pathology results consistent with dehydration and renal impairment. At noon the same day, day 3, the clinical signs persisted in the high-dose females. The study director and clinical veterinarian made the decision to stop dosing all the high-dose females due to the severe clinical signs of toxicity. At this time, the study director created a protocol amendment to stop dosing the high-dose females and sent the protocol amendment to the sponsor for signature per the facility SOP. At the same time, the study director sent an email to the study technical staff alerting them to stop dosing all high-dose females. In the afternoon of study day 3 at 4 p.m., all animals except the high-dose females were dosed per the study protocol. The clinical condition of the 3 high-dose females continued to deteriorate, and these animals were euthanized in moribund condition. On study day 4, all animals except the high-dose females were dosed per the study protocol. The sponsor had signed and returned the protocol amendment to the study director, who then signed the protocol amendment and distributed it to study staff. All remaining animals were dosed per the study protocol and protocol amendment for the remainder of the study period. Male high-dose animals continued to show clinical signs of toxicity. However, the signs remained minimal and no signs were observed during the 14-day recovery period. No other animals showed clinical signs of toxicity. However, there was evidence of renal impairment in the clinical pathology assessments from the mid and high-dose males and females. All the remaining animals survived to their scheduled study termination, and there were no test article related findings at necropsy. However, in histopathology, it showed evidence of dose-dependent renal epithelial cell damage with CAS in the mid and high-dose groups. So let's take a moment and consider the information I've just provided regarding the study. In the study described, did a protocol deviation occur? I'll give you a few moments to decide on your answer for this polling question from the 4 options provided, and then we'll take a few moments to go over the answers. So as we discussed, the correct answer is C. A protocol amendment was created but not signed by the study director prior to the day 3
Looks like we gave away the answer there before we saw the polling questions, but it looked like as I was watching the results come in, it looked like most people. Oh, there it is. let's go ahead and broadcast the results. So it looks like most people have the opinion that, yes, indeed, the change in dosing was not covered by the protocol amendment. And if that was your answer, that's great, because as you recall, the dosing of the high-dose females was stopped after the day 3 morning dose. The protocol amendment was created around noon on that day 3. However, it was not signed by the study director until day 4. So that protocol amendment was not in place when the high-dose females were not dosed on the afternoon of study day 3 and morning of study day 4. And even though the study director did alert the technical staff of the decision, and the decision was certainly valid based on the clinical signs of toxicity and for animal welfare reasons, the fact that the protocol in place at the time, and that's key there, the protocol in place at the time of the day 3 afternoon and day 4 morning dosing was not followed. And that created the need for the protocol deviation. So remember, it's the protocol that's in place at the time of that activity, and that's the key there with this particular situation, because at the time that they were not dosed, that protocol still indicated that they should have been. Now, obviously, for toxicity and animal welfare reasons, no one would disagree that they shouldn't have been dosed, but that doesn't have an impact on whether or not a deviation from the protocol in place occurred. So now let's go to the next slide, and we'll see the rest of the story.
PM dosing. The protocol amendment was not in effect until signed by the study director the following day. Therefore, a deviation from the protocol in effect at the time of the day 3 p.m. dosing and day 4 a.m. dosing occurred, and a protocol deviation should have been written to address this issue. In this case, the observation cited was that the quality assurance unit failed to determine whether any deviations from approved protocols or standard operating procedures had been made without proper authorization and documentation. And the reference, of course, is 21 CFR 5835B5. And now I will turn the presentation over to Dr. Zhou Chen, who will discuss case studies regarding translation and report accuracy.
Thanks, Lynda, and good morning. This is Zhou Chen from CDER OSIS, Division of New Drug Study Integrity. I will cover the second case, study report translation. Every week, CDER receives several hundred non-clinical safety studies, and more than half of the studies are from foreign countries. Many of them are from non-English speaking countries like China, Korea, and Japan. From our review and the inspections, we found that the report translation practice is not consistent among different CROs, and the CDER review divisions have concerns related to the accuracy of translated study reports. Many times, we found English translation of final study report does not accurately reflect the raw data reported in the studies. We have seen misspelling, typographic errors, inaccurate translations, paragraph missing, amended report missing, mislabeled parameters, wrong tables, formatting errors, and many other mistakes in translation. Some foreign firms had no idea that safety studies submitted to FDA should follow FDA's Part 58 GLP regulations. They follow their own country's GLP regulations, and as a non-OECD member country, CROs there follow OECD GLP for studies that were submitted to FDA. We also noted inconsistency in study director's signature and dates in translated study reports. We have seen translated study reports that are not scanned and dated. We have seen translated study reports with the study director's name printed on the translated version. And we have also seen translated study reports that the study director signed and dated with different dates, either original study finalization date or report translation date. We found in some foreign CROs, English proficiency is very limited for study director and the QA staff. So it is hard for them to verify the accuracy of translated report. Some firms had no SOPs for English translation, or they had SOPs but they never followed. In some CROs, translation was done by bilingual staff. Like they can hire a translator, so in this CRO, his job specifically is to do the report translation. In some CROs, translation was done by a study director, and some other firms, they contracted the translation out to a third party, or even to a friend, and their study director and the QA were not able to check the accuracy, so no check was performed. Talk about following FDA GLP regulations in safety studies. Some foreign CROs in non-OECD countries conducted studies following their own country's regulations. There could be several reasons, like the study may be originally performed for their own country's submission. Some CROs in non-OECD countries do not know that studies for FDA submission should be performed with compliance of US FDA regulations. Also, there are some reports the original version stated their country's regulations, and the English version stated Part 58 regulations. Studies performed in non-OECD countries should follow FDA Part 58 regulations. CDER review divisions sometimes ask OSIS inspection background for their decision making. Here is the deficiency 1. Inaccurate data and error with the translated report. This example is from a foreign GLP inspection. In this inspection, we compared the foreign version of several study reports with the English version and found many times the original language version is correct and the English translation of the final reports did not accurately reflect the raw data reported in the original version. There were many errors like missing words, mislabeled parameters, wrong tables, and spelling errors. The firm explained to us that they contracted out the translation to a friend, and they trusted this translator, trusted this friend, so QA and QC check was not performed. Okay. This is the 28-day dog study, the original version table 15 for clinical chemistry. It looks good, right? There are 6 parameters with different units. We can see only calcium unit is millimolar per liter. Here's the table 15 from English version of the report submitted to FDA. This poor quality was from the report, not from my slide preparation. This seems that this table was not finalized with right color words. We can see there are 7 parameters versus the 6 in the original table, with the addition of the CK and B. The column of calcium appeared twice. The values in the last column was copied from the calcium column, so the values were wrong, were not correct. The units were also wrong. There are 5 millimolar per liter units, but the original version had only 1. This is from the same report, the original version of table 17. Okay, and the hematology, it looks nice, the table looks nice. There are 9 items. All items are red blood cell parameters, WBC parameters. And the unit for the last 3 items is percentage. This is the English translation of table 17. The table had 10 items versus the original table's 9 items. We saw parameter errors, like the last 4 items are red blood cell parameters. But in the original table, there were no red blood cell parameters. All parameters are red blood cell parameters. Okay, and also we saw unit errors. There are 5 percentage units in the wrong place. Also, the values in the last column are the same as the column for the red blood cell count, the first column. So the values were wrong. So my feeling is that these tables are not finished during the translation. This is the translation-related Form FDA 43 observation we issued to the firm. The quality assurance unit failed to review the final study report to assure that such report accurately described the method and the standard operating procedures, and that the reported results accurately reflected the raw data of the study. Specifically, the English translation of the final reports does not accurately reflect the raw data reported in the original language version of the report. The study director is also responsible, as he did not ensure accurate data were included in the study report. Study director and QA could not check the report because of the poor English proficiency. However, at least I think they can compare tables. These are 2 other translation-related 43 observations issued in another inspection. First one, corrections or additions to a final report shall be in the form of an amendment by the study director. Specifically, report amendments were not translated. So in the original version, there are report amendments, but in the translated version, the amendments were not translated. Okay. Second observation, a final report shall be prepared for each non-clinical laboratory study and shall include the signed and the dated report of each of the individual scientists or other professionals involved in the study. Specifically, signed authority report not attached to the translated final report. Here we have another translation deficiency. Translation deficiency 2. The adequate handling of signatures and dates. Study director and KU signatures and dates left blank. Compliance statement, signature and date left blank. This case is from a CDER IND submission. In the original study report, the approved page was signed and dated by study director on February 22nd, 2017. But in the translated approval page, all names and the date, blocks were blank. Things happened to the compliance statement and the QA statement. The original language version was signed and dated, but the signature and the date blocks in the translated study report are blank. Deficiency 3. Inadequate handling of signatures and dates. Study director and the QAU signatures and the compliance statement dated when the report was translated. In the translated study report, the study director's name and date should be original report publication date, not the translation date. Here we have an example. We found in a 4-week monkey study was finalized on December 31st, 2013. But the translated English page, but in this translated English page, study director, QE, and facility management signed their names and put the current translation date. It's July 21st, 2016, instead of December 31st, 2013. Deficiency 4. Compliance statement implies compliance with Part 58 GLP regulations. For several CROs, the final report with the original language stated that their own country's GLP regulations were followed for the study, and no Part 58 regulations were mentioned. But in the translated final report, they added Part 58 regulations in the compliance statement. Here I have several questions for you to consider. The first one is who should do the translation? Should the firm contract the translation to a professional translator, or they can ask a study director or QU to do the translation, or to ask a back staff to do the translation, or can they ask a friend to do the translation? Second question, is a certificate of translation or a statement from the translator about the translation needed? So do we need a certificate or statement from the translator for this translated study report? Last question, what do the dates and the signatures mean on the translation of the study reports? And I can give you a few moments to think about this, and I will come back later.
Hi, this is Zhou Chen. Very sorry for the poor background quality. And now I just want to very briefly summarize what I just talked. I introduced the full deficiency in translated study report. The first case is we went to the firm, and we were able to compare the original study report in the original language versus the English translation. And we found the original version of the study reports are okay, but the translated version, there are so many mistakes, the omissions, and unfinished paragraph, and wrong tables, wrong values, this kind of thing. So we check with the firm. The firm said they contracted this translation to a friend. So they trusted this friend. So the study studies are really poorly translated, and the firm's study director and QA, their English level is limited. They could not perform the QA on the translated reports. So these poor quality reports were submitted to CDER. So you can see the these kind of reports cannot be used to support the clinical safety. Like the data were not considered accurate, and the firm is also very frustrated. They are surprised that they did not know the study report was so poorly translated and not finalized even. So we issued a 43 for this firm for study accuracy. And the second case is the study report translation. Study report translation in the original study report, the study director signed his name and dated the study report, but in the translated version, the signature and the date blocks are blank. And the third case is also the signature and the date blocks. The original version of the study director signed and dated the study finalization date, but in the translated version, they put the date. Study director signed the translated version, but they put the date as the translation date instead of study finalization date. Okay, and the last case is that the studies, the GLP studies, were performed following their own country's GLP regulations, but in the translated report, they added Part 58 regulations. So this is not acceptable. It's not a true translation. So here I have several questions for you to consider. The first one is who should do the translation? So from our first case, we can see not everyone can do the translation, right? So the first case, the firm had no idea the translation in several study reports was not complete, and they were sent to FDA, and we are able to compare the original report and found that the quality was very poor. So basically this kind of study data cannot be used to support the safety of the clinical studies. I listed several selections, like a contract to a professional translator. It happened some CROs, they have a translator. His job is specifically to do the report translation, And sometimes they have the study report or QA performs the study translation. And like the last case, they contract to a friend, So basically, I think should be the qualified translator to do the translation, And also here the second question is the certificate and translation or statement from the translator about the translation needed. Okay, so we have seen different practice, and the FDA reviewer have concerns for the quality of translated report. So if a translation certificate is needed, so the question here is how can you convince the FDA reviewers or show the translation is valid? And so our consideration is that we should have, if possible, there should be a certificate of the translation or statement with the translated study report, indicating that the study report is a true representation of the original documents, and the translator can sign his name in this statement and put the translation date in this statement. Okay, and next one is what do the dates and the signatures mean to the on the translated study report? Okay, same thing. So I think the translated report should be a true representation of the original study report. So the study director should be the name should be the original study director's name should be typed here, and the date of the translated in the study report should be the original study finalization date, not the translation date. You know, this is my consideration. Let's see. I'm reading some participant chat. You know, and answer to question 1: who should do the translation? Yes. So our consideration is that we don't have a standard answer for this one, but the answer is that the translator should be a qualified translator, you know. And also, I just want to also mention 2 other things that for the translation, there should be a SOP in the CRO. You know, in this CRO should have SOP, the SOP for translation, and this SOP should be followed. And the other thing is the non-OECD countries, like foreign countries, if they are non-OECD members, when they do studies for FDA submission, they should if the study was performed for FDA submission, they should follow the FDA regulations. Okay, let's read some comments. Okay. Okay, so let me see. Amendment. Okay. There's a comment to say translation should be validated from original language to the intended accepted language. I thought there are so many comments for the translation. Okay. So I think that's very good. So let's go back to the presentation, and I'll show you our expectation. You know, right? I'm also thinking, you know, maybe a guidance for the translation maybe is necessary. Okay, I think we can go back to the presentation. Thank you.
Okay, here are our expectations. Our current practice for foreign language translation is to verify that the CRO performing the GLP studies should have a SOP for study translation, study report translation, and the SOP should be followed. Translation should be performed by a qualified translator. We prefer to have a certificate of translation with each translated study report. The study report should include a signed and dated statement from the translator indicating that the English version of the study report is a true representation of the original document. Translated study report should not contain signatures. Instead, the translated report should contain the type names for study director, QA, and facility management, and the typed dates of the original study finalization by the translator. That is, translated report should include a signed and dated statement from the translator and the typed name of the study director and the typed date from the original study report. The translator should enter the names and the original report finalization date, not the translation date. So review divisions may request the original documents in native language and the translated documents. The review division should reject poorly translated document and bring them to the OSIS GLP team's attention. We may schedule a follow-up inspections later. Here are documents we use to guide our GLP inspections. Part 50 regulations and the GLP compliance program. In summary, OSIS serves as one of the last flight of eyes for authenticating data and to provide assurance that the data supporting regulatory decisions are reliable. The case studies presented today highlight two of the common non-compliance observations from GLP studies. Failure to identify and issue protocol deviations when required, and the issues observed in English translations of final study reports from GLP studies. GLP regulations provide the framework to ensure the quality and integrity of data from non-clinical studies, so any non-compliance may impact the quality of the data submitted for review. Valid non-clinical safety data are essential to hazard identification and risk assessment for clinical trials. That's the end of our presentation. Next, Lynda and I, we are happy to answer questions. Thank you.
And welcome everybody. We're looking at some of the comments that came in during the chat for any final questions. Right now, Zhou Chen and Lynda Lanning are looking over the comments and everything you had on both case 1 and case 2 so that they can provide some final thoughts and answer any questions before we wrap up the session. So hold on 1 second and Lynda and Zhou will be right with us.
So thanks everyone. Excuse me. Thank you everyone for your questions in the chat box. Hopefully, I think I've been trying to keep up with them for the most part. Excuse me again. So hopefully I've been able to answer the questions. So I think a lot of the questions that I've seen come through are the different, are trying to understand the difference between a deviation and an amendment. And in the case of this study, the deviation, as I reiterated during the polling question, the issue here was that the protocol amendment had not been signed by the study director and therefore was not effective at the time that the particular study activity, which was animal dosing for the high dose females, occurred. So you can see that the importance here is when that amendment is signed with respect to when that activity is occurring. If the protocol amendment is signed before that activity occurs, then the protocol amendment is in effect, and the activity occurred with the protocol that is in effect at the time. So there would be no deviation then, because it was done within the constraints of that study protocol amendment. If the activity occurred and the protocol amendment was done after that, then there's a protocol deviation. So it's about the timing of when those two events occur, when the amendment is signed and effective, and when the study activity occurs. So hopefully that gives you some more clarity around that. So I think Dr. Chen is available. I know there are a great number of questions. Okay. All right. Thank you so much, everyone.
Great. Yeah. Hi. I saw so many questions, and I saw some like I saw Stacy answered some questions. Thank you, Stacy, and you are correct. And basically, there are a lot of questions for the translator. Who can be the translator? Some people said I didn't answer that question. So very unfortunate right now. FDA has no FDA does not have a requirement for the translator, But from our inspection, we found so many poorly translated reports. So we really think the CRO should have a qualified translator, So as I mentioned, we are thinking about guidance, for the report translation, and yeah, and also some people ask the SOP, for translation. Yes, some CROs they have SOPs for translation. So basically, we cannot inspect a translator, but we can audit the SOP to see if the CRO followed their SOP, and if they have an SOP, and if they follow the SOP, this is what we can do, Yeah. And let me see other questions. I think, Lynda, do you have anything to say?
No, just that I think, as just to reiterate what Dr. Chen was saying, and the FDA expects the translation to be an accurate translation of the report. So that's really the one of the bottom lines there.
Right.
The takeaway message for that.
Yeah.
Thank you.
Yes, and also one big issue is that some CROs, foreign country CROs, they follow their own country's GLP, and they send the studies to FDA. This is the big concern. CDER reviewers many times they ask us for the background of the CRO and ask the study quality. So I just want to mention that foreign CROs, when they prepare studies for FDA submissions, they should follow FDA GLP regulations. Yes, and also study report translation. They should type. We prefer they type the study direct name and type the original study finalization date in the translated study report. Yeah, and today I just showed you our expectation for the translation. Thank you very much.
All right, looks like that's all the time we have for our first case. Thank you so much to Dr. Lanning and Dr. Chen for a great case, and thank you all for the participation. We'll move on to the next case in our section.